Welcome to Learn with Sam. Let's continue with the genetic terminology. So the first terminology is F2 generation, the second generation progeny produced by selfing. Inbreeding of F1 generation offspring is called a second filial generation. Okay, now let's take a look at this with an example. So as you can see here, parental generation are two parents, homozygous parent with contrasting character, capital T and small t. So when we cross them, we'll get our F1 generation with genotype capital T and small t. So to get F2 generation, we have to self-pollinate F1 generation, or we have to inbreed F1 generation. What is inbreeding? Take two F1 generation offspring and cross them. That is inbreeding. Okay, so either we have to self pollinate F1 generation or we have to mate to F1 generation offspring. So after mating or after self pollination of F1 generation, we'll get our F2 generation with genotype capital T, capital small t, capital T and small t. As you can see, F2 generation has both the characters, parental character. F2 generation has both the genotype of the parental generation, that is capital T and small t. These are the genotype of the parental generation. So the next genetic terminology is homologous chromosomes. The morphologically, genetically and structurally essentially identical chromosomes present in a diploid cell are called homologous chromosomes. Each cell in the human body has genetic material DNA which is coiled to form the chromosome. So in human cell there are two set of chromosome. Okay, and they both are structurally, morphologically, that is morphologically, genetically and functionally identical to each other. Next is back cross. It is a cross of F1 progeny with any one of the parents. So when we cross F1 generation with any one of the parental generation, that is called as back cross. Now test cross. Test cross is a cross of F1 progeny with homozygous recessive parent. The main difference between back cross and test cross is, in back cross we will cross F1 generation with any parent. It can be homozygous dominant parent or homozygous recessive parent. Whereas in test cross we have to cross F1 generation with homozygous recessive parent only, that is the parent with the recessive genotype small t. It is used to test homozygous or heterozygous nature of hybrid. So test cross is basically done to check if the parental generation is homozygous or heterozygous. It is a kind of back cross. Now let's look into this example. So the parents are homozygous parents with capital T dominant parent and small t recessive parent. F1 generation is capital T small t, and when in test cross the F2 generation for the F2 generation, capital T small t F1 progeny has to be crossed with the recessive parent, that is small t small t. Next genetic terminologies are phenotypic and genotypic ratio. First is phenotypic ratio. It is a ratio of offsprings produced in F2 generation and subsequent generation with respect to their physical appearance. So in F2 generation, in a monohybrid cross, that is when we cross two parents for only one character, we'll get 3 tall and 1 dwarf plant. So the phenotypic ratio is 3:1 in F2 generation. Whereas genotypic ratio is a ratio of offsprings with respect to their genetic constitution. That is, in F2 generation we'll get 1 capital T, 2 capital T small t and 1 small t. So the genotypic ratio is 1:2:1 in F2 generation for monohybrid cross. Now let's see the difference between test cross and back cross. So the first point of difference is, test cross is a cross that occurs between dominant phenotype and a recessive phenotype. Back cross is a cross that occurs between generation F1 hybrid and one of the two parents. So in test cross we have to cross F1 generation with homozygous recessive parent, whereas in back cross F1 generation can be crossed with any parent. It can be homozygous dominant or homozygous recessive. Second point, test cross is breeding of dominant phenotype with its recessive phenotype. As mentioned earlier, in test cross we will be crossing one dominant character with a recessive character, that is dominant phenotype to a recessive phenotype. In back Whereas in back cross, the F1 hybrid is either crossed with the dominant parent or it can be crossed with recessive parent. Third point, all test cross are back cross, because in back cross is what F1 generation will be crossed with either dominant or recessive. So if F1 generation is crossed with the recessive parent, it is back cross. Whereas all back crosses are not test cross, because in test cross only F1 generation has to be crossed with recessive parent, but for back cross it can be crossed, F1 progeny can be crossed with dominant parent also. Test cross identifies the zygosity of the dominant phenotype. Back cross recovers the elite genotype. A test cross is important to determine the genotype T or small t of the dominant phenotype. Backcross is useful in recovering important characteristics of the parent. So a test cross is done to check whether the parents are heterozygous or homozygous, and a backcross is done to recover some important genotype. Thanks for watching. Do subscribe to our channel.